Hi, all of you. Welcome to part 4 of the cardiac chapter. We start out by looking at a review of anatomy and what a cardiac contractile cell looks like. And it actually looks different from the skeletal muscle cell, and these are some of the differences. If you look at it here, one thing that you notice is that the cells can branch. So you have one cell here, and it's sort of branching in two directions. You don't see that in skeletal muscle. The cells are connected by intercalated discs or gap junctions. Remember the intercalated discs? These discs, we had terrible time finding them under the microscope in anatomy. They are a gap junction that's going to allow, very important, it's going to allow those cells to be electrically coupled. So I no longer need neurotransmitter to go, for example, from here to connect to this cell. What's going to happen instead is they're electrically coupled. So if I have an action potential on cell number 1 here, I'm going to have it on cell number 2 because it's going to travel right on through that gap junction without a chemical, without a neurotransmitter. Okay, we say those cells are electrically coupled. You want that because in the heart you need to have this radiating action potential that's going to result in a contraction. And it needs to be very coordinated. And that coordination is going to come from the fact that these cells can communicate with action potentials directly and depolarize. Okay, let's look at molecular similarities between cardiac contractile cells and skeletal muscle cells. As a review, here are some of the things that were similar. When we think of them depolarizing, that depolarization is triggered by voltage-gated sodium channels. So a sodium channel is going to open. You're going to have an action potential along the surface of that muscle cell. No different now. Yeah, you're going to have that action potential traveling down to a T-tubule. No different. It's going to arrive at the sarcoplasmic reticulum. No different. There's going to be a release of calcium from the sarcoplasmic reticulum. This should be sounding super familiar by now. These are the steps that you learned when you think of skeletal muscle. And then some of that intracellular calcium is going to trigger cross-bridge formation. You're going to have actin sliding over myosin, and that cell is going to shorten. Right? You have the sliding of the myofilaments. And that's very similar in cardiac also, with some differences. And that's what we're going to look at. What are those differences? Molecular differences between cardiac contractile cells and skeletal muscle cells are one in the sarcoplasmic reticulum receptor. So when that action potential is coming down and hits a receptor, that receptor is going to be slightly different. And also in the role of calcium in the cell. So let's look at that with diagrams. I can show you this first diagram that's very busy and has a lot on it. So that's not going to work for us. I'm going to go on to the next slide and show it to you in pieces. Step number 1. Step number 1, you have an action potential that's arriving from a neighboring cell because you had that gap junction. So in 1, here it is in red. This action potential is traveling down the T-tubule now, and it's going to hit this particular channel. And this channel is called an L-type calcium channel. So that's different. You have a calcium channel there now. And it's called L-type. I'll tell you a little bit more about that here. And then I'll backtrack again. What's an L-type calcium channel? It's also known as a DHP receptor that we looked at before. It's voltage dependent, but it's a calcium channel instead. And the L is standing for long lasting, referring to the length of the activation. It actually stays open for a while. Okay, so let me backtrack now. We go to number 2. Number 2 says, voltage-gated calcium channels open and calcium enters the cell. So this is weird. This is different, right? I have this calcium channel and this calcium that was floating around in the cell here. I have lots of that on the outside, is now going to be allowed to enter into the cell. Okay, different. from skeletal. And then what's it going to do now that I have this entryway of calcium? Step 3, the calcium is going to induce a calcium release through a ryanodine receptor. So, okay, another different receptor now. Here this is the sarcoplasmic reticulum, and they're illustrating it on both sides. So once it hits the sarcoplasmic reticulum, it's going to stimulate the release of calcium. So weird that it's calcium stimulating the release of more calcium. And in step 4, what they're calling this is a calcium spark. So that's a weird name. It comes from when they photographed this in the very beginning when they were studying this, physiologists were studying this, they saw it like a little flash on their film. So they called it a calcium spark. I think of it more as the release of this calcium. Instead of being a fluid thing, it releases it sort of in pieces. So it's in bursts. This calcium spark is kind of like a burst of calcium, and we don't really understand it very well. The molecular mechanism of this is still obscure, so we're not sure what it is, but think of it instead of burst of calcium. That's what's going to happen. Okay, I'm backtracking. So I have calcium coming in. It's stimulated this receptor, the ryanodine. It's going to release more calcium in sparks. So I have spark 1, spark 2, spark 3. And when I have enough of these sparks that are accumulated, we're summing up that volume of calcium that's entering that cell, then it's going to be sufficient to cause a signal. A signal not that different from what you looked at before. That signal then is going to allow that extra calcium will be able to attach onto troponin. It'll move tropomyosin out of the way. Myosin will be able to attach onto actin. From then on, the story repeats itself. It's very similar. Okay, so backtracking again. Step 2 seems different. Correct? My channel seems a little different. And sparks seem different, and that's what is going to set it apart from the skeletal muscle contraction. Okay, over on this side here, I'm going to show you after this contraction is over. This drawing is going to show you what happens to put it all back together again. I call it sort of Humpty Dumpty back together again, because here we've released calcium. There's all sorts of things happening. We're going to then remove the contraction and create a relaxation. So we'll draw it on this side. If you had that muscle here, and now you need to relax it because this contraction's over, you have the lub-dub happening, right? Now it's going to relax, and myosin is going to disengage from actin. You see it here, that it's no longer attached. And it's going to release that calcium. So step 7 is relaxation occurs when the calcium unbinds from the troponin. And it unbinds, and it's now floating around in the cytoplasm of the cell. What next? Where to put it now? Because you don't want it there because it would cause another contraction. So calcium is pumped back into the sarcoplasmic reticulum for storage. When you use the word pump, you know that you mean ATP also. So we're using energy, we're using ATP in step 8 to put that calcium back into the sarcoplasmic reticulum. That does not sound so different from skeletal muscle again. What next? Well, we can't store that much calcium in the sarcoplasmic reticulum, and we need some of it outside here for the next incoming contraction. So we need to get some of that calcium out of the storage unit and back out into the extracellular fluid. How so? We've put some in storage. The other then, what we're going to do is get it out. step 9, calcium is exchanged with sodium by an antiport. So this antiport is, okay, I will pump calcium out, 1 calcium out for 3 sodiums back in. And this is the NCX antiport. Sodium in, calcium out, and it does not use energy. It does not use ATP because it's doing that antiport mechanism where one slips in with the other one. And this is down its concentration gradient. All right, that's 9. Let's go to 10. Once I have all this sodium, too much sodium back inside the cell again, that's not good. I need to get it back out again because now I have an excess of sodium inside the cytoplasm, and that's not homeostasis. I need to remove it. So there is a second antiport here, which is the good old sodium-potassium pump, and that does use ATP. So for every 3 sodiums that are pumped out, 2 potassiums are pumped back in again. And that's sort of the end. So you can imagine yourself back here again with another action potential. I now have enough calcium for this again to trigger another calcium release and a spark. And this sort of mechanism continues happening every time you have lub-dub, lub-dub, right? Contraction, relaxation, contraction, relaxation. This is the last slide of this video. The question is, can cardiac muscle contractions be graded? So graded contraction was one that was a partial contraction that wasn't a complete contraction. It was just a contraction in skeletal muscle that maybe contracted just a little bit and then released. It's a contraction maybe that didn't even make it to the axon hillock, remember? Well, here, more depolarization produces a stronger contraction. Just imagine that the heart at times does have to contract more forcefully, correct? So that is going to happen. More calcium is going to enter the cell, increasing that cross-bridge formation. If you have more calcium in there, then myosin can stay attached onto calcium for longer. If it stays contracted on for longer, then it's going to be able to contract more forcefully because it will be able to slide more. So this we think of always doing sympathetic stimulation. The sympathetic is going to speed up that heartbeat, and the parasympathetic is going to slow it down. So doing the sympathetic stimulation, there's going to be an increase in the permeability to calcium ions. So all of a sudden, those calcium ions are coming in quickly. And therefore, that's going to result in an increase in contractile strength. I hope that's clear. Thanks for listening. If it's not clear, make a note of it, and then come to a Zoom session, a review session. Thank you. Bye-bye.